All right, this video is for part 1 of chapter 10 on antimicrobial drugs. We are going to cover in this first set of slides the broad overview, which actually will take the majority of the time here. So we'll talk about concepts and new vocabulary terms surrounding this concept of antimicrobial drugs or antimicrobial medications, and then specifically look at each of these different major classes of drugs and the 5 different sites of action, so where on the bacterial cell these drugs affect. Now this chapter is all about antimicrobial drugs for bacteria. I guess classically we'll term these things antibiotics, but note that there are antimicrobial drugs for any type of microorganism, so antifungals, antivirals, even though those aren't live organisms, but still pathogens, parasitic medications, right? So keep that in mind. Even though we're not talking about any of those ones, we're focusing on antibiotics that impact bacteria. Just that this is a one small class. So this first set of information, these terms, this is really applicable across the board. So when we use the term when we use this term chemotherapeutic agents, and this is sort of from the most broad category down to the more specified. Chemotherapeutic means chemo is for chemical, therapeutic means treatment of, right? So something that actually impacts a disease state, and whether that's a disease, an organ disease, etc., or an infectious disease, it doesn't matter for chemotherapeutic agents. So this is any type of chemical that's used in the treatment of any kind of disease, right? Chemotherapeutic drugs are used, you guys hear this term a lot when talking about cancer. So chemo, that kind of abbreviation that we use, that slang term, it's talking about using a chemotherapeutic agent to target the rapidly proliferating cancerous tissue. But chemotherapeutic agents are used in infectious diseases as well. So an antimicrobial drug, specifically anti, of course, is against microbe, any type of microbe. In this case, it's a chemotherapeutic agent used to treat microbial infections. And I will say antimicrobial drugs or antimicrobial medications, this is really the most commonly used term today. I mean, if you go to your physician, they'll still talk about antibiotics, but in the scientific realm, when we're talking about these medications, these chemotherapeutic agents that are used to target bacteria, viruses, fungi, really we are shifting more towards the use of this term antimicrobials or antimicrobial drug instead of antibiotic, because it's more a more applicable and didactically correct term to use. No, that's a weird phrase, but. And then this actual term antibiotic, what it really means is an antimicrobial drug that is naturally produced by a microorganism. Now, antibiotics were really how this whole thing started, with antimicrobial drugs for antibacteria. So things that antimicrobial medications that specifically target bacteria. The first, of course, that was discovered and really turned into human use is penicillin back in the early 1940s and 50s. This is naturally produced by the penicillium fungi. And so therefore it is truly an antibiotic. It's a naturally occurring antibacterial drug that is produced by a microorganism that we essentially just grew a bunch of penicillium fungal organism and extracted out that chemotherapeutic agent and then purified it, and now we have antibiotic, a drug. Today, most of the antibiotics that are on the market, not all, but most of them are synthetically produced. So even penicillin, even though originally it was an antibiotic, something that was produced naturally by a microorganism, today often it is synthetically generated. So really it's technically shifted back to just an antimicrobial or antibacterial agent. So just to get technical on these things, I'll try to use antimicrobial drug or antibacterial drug when we discuss this stuff, but I'll probably still slip back into the use of this term antibiotics, since that has been sort of the history of this whole class of treatment for bacterial infections. So things to consider when choosing a drug. We're going to spend a little bit of time on this category because this is where we get the conceptual understanding for this information. So I understand that most of you in taking this course are going to be going into nursing, or some people go into pharmacology, some people go into veterinary science. And very few of you will be PAs, and even fewer of you will eventually go on to be a physician. And those are really the only two types of people that would go on to choose an antimicrobial drug to administer to a patient. So I understand that. Let me just say that first and foremost. Nonetheless, anyone in healthcare, anyone in patient care whose patients will be receiving antimicrobial medication to treat infectious agents really do need to have a very good understanding as to the conceptual basis for choosing these different drugs. So it's not as easy as administer a broad spectrum antibiotic and send them on their way. There are lots of other things to consider when choosing or selecting for the best antibacterial drug, antimicrobial medication. So these are some of these things here are just some basic things to consider. Ease of administration. So ease of administration really has to do much more with the patient and what we're going to term patient compliance. Thing over. So what does this mean? Compliance, right? How do you get your patient to take the drug? Most of us have experienced at least one bout of antibacterial medication that we've had to take ourselves. And so you should be familiar with the requirements. Usually these are, you take 1 to 3 pills per day for 7 to 10 days, right? And you must complete the whole course of antibiotics or antimicrobials in order for them to be truly effective and to not develop resistant bacteria. So how do you get the patient to comply with this? And again, just thinking about anecdotal, so firsthand knowledge or your own story, just think back to when you took that course of antibiotics. In almost every case, except for when the infection is not able to be treated well by that antibacterial medication, in almost every circumstance, as long as you have successful treatment, the patient feels fine, feels much better after about halfway through that antimicrobial medication treatment. So again, going back to patient compliance, how do you get that person, even when they feel great, after 4 days, 5 days, and they still have 2 more or 5 more days of medication to complete? How do you ensure that they complete that full course of antibacterial medication to reduce the development of the more resistant organisms? One of the ways is, one of the things to consider is ease of administration. This is how you are administering, how you are providing this medication to the patient. Are you doing an injection? Which of course would be really important if your patient is having a severe infection and is hospitalized, right? But what if your patient is at home? If you're providing medication and it needs to be injected, is that likely to occur? Probably not, right? There are very few circumstances that an injection, IP or IM injection or IV injection would ever be done at the home. And so consider the ease of administration. So injection, of course, would be one. Oral is the most common, which is easier to take. Topical would be another, which is even easier sometimes to administer, right? So how are you getting this medication into or onto your patient? The second concept here is penetration. Penetration, we talked about in our last chapter, is being important when you're choosing an antimicrobial control method to make sure you are penetrating the thing that you are trying to clean, like fibrous tissue. Make sure it actually penetrates through the fibers to get down to where those bacteria, viruses might be hiding out. For antimicrobial medication, penetration means like make sure the medication gets to the site of infection. Again, if you're thinking about how you are administering this, if you are providing something orally for, say, actually, like I was just thinking about this 2 years ago, my daughter had a Staphylococcus aureus infection in her toenail. It was kind of gross. And the physician administered an oral broad spectrum antibiotic or an antimicrobial. So how easy is that oral medication going to get all the way down to that really external part of the toe, right underneath the toe, or right in that toe bed, toenail bed? And is it going to get there in high enough amounts or high enough concentration to have effectiveness there? This next one, toxic to agent, non-toxic to host. This should be very self-explanatory. You definitely want something that is very selectively targeted, right? And so we actually call this selective toxicity. And you want this, definitely. You want it to be selective only to the bacterial agent and not to the host. And when we use this term host, we're actually not just thinking about the human, but remember, what else do we have in us? Normal flora. So now adding complexity to this issue, right? We understand from our chapter 3 and chapter 4 how to differentiate between human cells and bacterial cells. That's not too hard. But now we have to selectively differentiate between the bacteria that are the pathogens and the bacteria that are not, right? So this becomes really important, and we'll give us a new term in just a second. And then stay active in the body. So your body is really great at clearing anything foreign that comes into it, and especially antimicrobial medication or chemotherapeutic agents. So we have a really good filtration system, and our liver clears things out very, very quickly. So this is something to keep in mind. This is why if you are in pediatric medication, it's important to get the body weight of the patient before determining the concentration of that bacterial medication or chemotherapeutic agent to provide, because it really has to do with maintaining a high enough concentration that is targeting or effective towards the agent, but not toxic to the host. Okay, let's look at each of these things and break them down a little bit more. We'll also get some new vocabulary terms for you. So going back to toxicity, and actually I'm gonna give me just a second. So to better discuss toxicity, I wanted to bring up this concept of the therapeutic index. The idea here is for a drug to be selectively toxic, to harm the pathogen and not harm the human, right, the host. So the concept of the therapeutic index, this is a measure of selective toxicity, and this is done with any chemotherapeutic agent out there. That's from cancer drugs to antimicrobial medications, right, to really anything, any kind of drug that a human is taking. And so to measure the selective toxicity, or to determine the therapeutic index, what you're doing is plotting out the therapeutic effect. In other words, as you increase the concentration of the medication, then you know, hopefully, that this medication, and let's just do this as an antibacterial. So let's say you have a bacterial infection. You know that in order for that bacterial infection to be cleared, you need at least to get a high enough concentration that it is effective against the majority of the pathogens in the body. So this term ED50, ED stands for effective dose. Okay, so the effective dose in the case, whoops, ED is the effective dose. So effective dose against at least, or in at least 50% of individuals, or to clear 50% of the pathogens, or whatever your 50% target is, right? It's the most effective dose. Now for microbial medications, antimicrobials, what we're trying to determine is the dose that it becomes toxic to the pathogen. And we'll talk about this in a little bit at the end of these next set of slides, but we can also use the minimum inhibitory concentration, or MIC, to determine the dose that it becomes toxic to the pathogen. So ED50 is the classic use of the therapeutic effect to determine the concentration needed for it to be effective in 50% of individuals or against 50% of the pathogens, or the minimum inhibitory concentration can be used here. Whatever it is, if you just kind of imagine this blue arrow as the increasing therapeutic effect of that antimicrobial medication as you increase the concentration. Now, for any chemotherapeutic agent, there is either targeted or off-targeted effects, so some type of toxicity to the human. For anything, if you increase the concentration of anything, even a food, right, then it will eventually become toxic to the human. For chemotherapeutic agents, this is a lot more usually so than a food item. So you can plot this out too, and these are graphs that are created usually from the clinical trials for these things. So the graph in red would be something that from a clinical trial or from case studies in humans, this toxicity of that chemotherapeutic agent to humans would be established. So this is the toxic effect on the human population, not the therapeutic effect against the disease or the pathogen, but now the toxic effects on the human. And so this would be established, right, from trials where you would show that at increasing concentrations, there begins to be some toxicity, either direct or indirect toxicity to the human. And again, the number that would be plotted would be toxic dose 50, right? So in 50% of the population, that's when you have essentially toxic effects that are evaluated or that are evident. So here for our therapeutic index, this index would be a ratio of the dose that is toxic, it's a little bit windy out here, the dose that is toxic to humans, or the TD toxic dose, 50 and 50% of the population. Toxic dose divided by the dose that is toxic to the pathogen, or the effective dose 50 with a minimum inhibitory concentration. And so what this index is actually evaluating is this number here, right? That's what you're looking at, is the difference between where it's toxic to the pathogen and where it's toxic to the human. And the wider this gap, the better that drug would be to administer in the human. So if you just kind of think about this, that means that if you chose this dose, where the therapeutic effect is at about 50% of that population will have that therapeutic effect. So it would be effective in 50% of the population. If you chose this dose here, then actually that means that there is no toxicity to the human, right? If you were trying to get a therapeutic effect that's effective in 75% of the population, for example, right, that would be this concentration here. And although we're still kind of at the low end of this curve, it may still be effective. If you were trying to find the best possible concentration where you have the most therapeutic effect here, now you can see it starts to overlap. I know my curve is bad, but you'll see it starts to overlap with toxicity to the human. So the wider apart these curves are, in other words, the larger this therapeutic index, the more selectively toxic that therapeutic agent will be. Let me clear this slide. So here is an example of two different types of drugs, one with a very high therapeutic index and one with a very low therapeutic index, just again to reiterate these concepts here. So the higher the number, the better. That's kind of what we're getting at for therapeutic index for selective toxicity, to make sure that we are getting a therapeutic drug, antibacterial in this example, that is selectively targeted to the pathogen and has no toxicity to the human at the concentrations that are being used. So it's again, just look at these examples real quick. For this one here, this is a good drug, because you can see that at any concentration that you choose, right, from where you have any kind of effective dose to 100% effective dose, right, any of these ranges here, this concentration, there is no toxicity to the human. You would have to increase that concentration massively to begin seeing any toxic effects to the human host here. And the toxic effect, where essentially you would just not want to use this drug at all, is so far outside of even the maximum concentration needed to be effective against 100% of infected individuals or 100% of that pathogenic population. This gap is just so wide that really there's no overlap, right? So this is a really, really good chemotherapeutic agent to choose where there is no overlap whatsoever. In some circumstances, like in antibacterial, with bacterial infections with super pathogens, ones that are super resistant to many different antimicrobial medications, then you may have to resort to something like this. This is no good because at the lowest concentration needed to be effective against the pathogen, you already have toxic effects in some percentage in the human population. And if you're trying to optimize that antimicrobial medication to target, the majority of the pathogenic population, you are now having a really high rate of toxicity to the host, right? So anything that kind of overlaps at these two curves is really no good. And this would be measured by this therapeutic index between the ED50 and the TD50. So you can see that this number here is very small compared to this number here. This distance between this ED50 and this TD50 is a very large number, which indicates good. There's a very small number, which indicates no bueno. Okay, hopefully this makes sense to you. Continuing on with these different terms here and these conditions or characteristics of antimicrobials. These are new vocabulary verbs for you, terms for you. We did talk about bactericidal in our last chapter, chapter 9, antimicrobial control methods. So when we're talking about antibacterial drugs, we need to discuss whether the drug is bacteriostatic or bactericidal. Bacteriostatic medications, they just inhibit. So static is like stopping or freezing something. So these drugs inhibit the growth of bacteria, but they don't kill them. That means that when you remove bacteriostatic antimicrobials, then the bacteria will continue living, right? So as soon as you remove this inhibitor, then the population can resume. Versus bactericidal, kills bacteria altogether. So let's talk about these terms and when you would use one versus the other. I'm not a physician, so don't take my professional term words for this, but the conceptual understanding is this. If you have someone who is otherwise healthy, that means, of course, they're diseased because they have some kind of bacterial infection, but they are otherwise healthy. Then you want to induce natural immunity. And the development of memory, if we recall this from our chapter 13. By essentially kicking our immune system into gear. So bacteriostatic medications would halt the growth so that your immune system can respond, which is good. Anything that induces natural immunity when we have the development of our memory response is very good. That means that if we are introduced to that same organism the second time around, you would be able to respond quickly to it. If, of course, you're dealing with someone who is immunocompromised, and if you recall, the individuals who are naturally immunocompromised would be the young, right, infants, the old, and diseased. Disease means immunocompromised condition, autoimmunity, et cetera, right? There's lots of diseases that fit into this category. Or if you're dealing with an organism that is incredibly virulent, like necrotizing fasciitis, Staphylococcus pyogenes, something like that, that has a very, very rapid rate of growth, you probably want to use a bactericidal. bactericidal, right? You want to kill the organisms because in these individuals, they might not have a ability to mount an effective immune response. And therefore, when you remove the medication, if they are receiving a bacteriostatic drug, then their immune system has not effectively eliminated that population. And therefore, they could have a redevelopment of their infectious agents, infectious disease inside that person, right? Because they just have not mounted an effective immune response. So in these individuals, we want to just completely eliminate that pathogenic population altogether, ideally. Now, this last term here, these combination drugs, if two drugs essentially cancel each other out, we call them antagonistic. This is really important for people in healthcare, especially the first line of defense, the nurses who are going and getting oral history for our patients, et cetera. If you hear of any kind of medications that the patient is already taking and you know already that they are contraindicated with a prescription that the doctor is wanting to offer, that would be considered an antagonistic, right? Contraindication would be the same kind of concept here, where they essentially do bad for each other. They either cancel each other out, or in combination, they provide some type of toxicity to the human. So you definitely want to stay away from any of these kind of antagonistic combinations. Synergistic, however, is good. So this antagonistic is bad. Synergistic is good. I'll give you one example of a very common synergistic drug as we go through our list of drug categories, one that we've already evaluated in our antibiotic lab. That would be sulfamethoxycillin or sulfamethoprem. No, oh my gosh. Sulfa and trimethoprim. Now I'm just making myself silly. We should just call it sulfa, SFX, or sulfa and trimethoprim. Sulfamethoxycillin and trimethoprim. Those are synergistic drugs. Both of them target different parts of the folic acid synthesis pathway, which is important in developing like nucleotides and your DNA and RNA, et cetera. Or for bacteria, they make their own folic acid. So if you target that pathway, it can completely abolish any metabolic activity or reproductive capacity in those organisms. So that would be one example of a synergistic drug. Essentially, we provide drugs that target different enzymes along that folic acid pathway, so that it's like a double whammy, right? It gives you a greater impact on these organisms. Perhaps if they're resistant to one, then they won't be resistant to another. That's in part how the cocktail works for HIV, the retroviral therapy or heart medication. This is a cocktail of different antiviral medications kind of doing the same thing. They all work synergistically to combat viral replication cycle at different spots along the way for the same essential reasons, so that if that virus has mutated and is essentially resistant to one type of drug, then maybe another type of drug will work in its stead. This is a couple of other concepts here. So targeting that drug, this is kind of thinking about penetration, this concept of penetration, making sure that that antimicrobial medication reaches its target tissue. So some drugs can be just directly targeted. If you have a localized infection on the skin, of course, you can selectively target that tissue by doing a topical instead of an oral treatment. If you have an infection in one region of your tissue, then you can inject that medication directly into the infected tissue through intramuscular injection instead of oral, instead of topical, right? So these are things to consider depending upon the nature of the infection, the severity of the infection, especially the location of the infection, and the circulation of the blood at each of those different target spots. This becomes really important also for people who have cardiovascular issues, especially diabetics that have been diabetic for a long period of time. They've lost a lot of their circulation in their extremities and so are very prone to infections. If you provide them with an oral medication, then the same reason that leads them to become prone to infectious disease in their extremities is what's going to limit the therapeutic, the penetration of that tissue, which is the loss of circulation. So these things are really important to keep in mind when considering each individual type of infection. What type of tissue is involved? What's the nature of the infected individual? Are there other diseases or disease phenotypes that need to be considered? Dose determinant is another kind of concept here to talk through. So dose is oftentimes determined through the half-life of any given medication. Let's talk through this one too. So dose, there's kind of a couple different things to think about here for dose. We were talking about the frequency, right? How often do you have to take this? Classically, you guys know 1 to 2 to 3 oral per day, right? 1 would be probably easier for patient compliance. 3 is a lot more challenging. If you've ever had a course of antibiotics where you had to take it 3 times a day, how many of you have actually completed that to the T? I'd be surprised if there were any of you who were able to complete a 10 or 7-day course of oral antibiotics that you had to administer 3 times a day. And the concentration, right? So in each of these administered oral tablets or whatever that might be, what's the concentration of that medication? So both of these concepts together make up the dose, right? How often you take this and what's the initial concentration inside each of those administered items, like the injection or the oral tablet. And this in large part is dependent upon the half-life, how long it takes for the body to eliminate half of the original concentration. So I told you already, your body is really great at filtering things out that you put in that are slightly toxic or at least foreign to the human body. So because of this known and FDA-approved antimicrobial medication on the shelf, right? Anything that you can purchase or put inside the human body has a known half-life. And this is due to actually a bunch of different conditions. So this is still sort of an average in 50% of the population. This also depends upon the weight of the individual, the body mass index of the individual, right? Which kind of is a reflection of their metabolic activity. If there are subclinical manifestations or even other disease conditions in the individual, or organ disease or organ failure, the half-life is going to vary. But this is sort of a general picture in half of the or in 50% of the population. How quickly does that drug clear out of the individual? So if you have something that has a long half-life, that means it takes a long time for that medication to be cleared, right? So a long half-life will require a low dose, because it stays in the body for a prolonged period of time. So for example, if you have something that you only had to take once a day, it likely has a very long half-life, which allows you to have low dose or low administration of this in the individual for it to maintain the minimum inhibitory concentration for that duration of the infectious disease. If it has a very short half-life, that means you need a high dose. Either a high concentration or administered frequently. Usually for things that have a short dose, you don't really want to mess with the concentration too much. If you recall back to our conversation about therapeutic index, the higher the concentration, the more likely that you're going to run into some toxic effects on the human. So if something gets cleared very quickly, you likely are just going to increase the administration of a low concentration oral tablet, for example, and just hope that your patient consistently administers that medication so that you maintain the minimum inhibitory concentration. And so I'm going to keep just bringing that back again. Really want to ensure the minimum effective dose or minimum inhibitory concentration in order for it to be effective against that pathogenic infection for the duration of administration or duration of the infection. The spectrum of activity here, this is going to be similar to what we talked about for our last chapter, but it's kind of inverse. For antimicrobial medications, remember we have a big problem with our selective toxicity in the human, which is our normal flora. Right? Ideally, we will not target the flora that is bringing benefits to us, and we selectively target only the pathogen. Ideally, you can get all the way down to species level targeting or specificity, but sometimes genus level is okay. So this is the spectrum. Broad spectrum is good for a wide range of bacteria, which has its benefits, of course, but it has major setbacks, which is the impact on normal flora. Narrow spectrum is ideal, but it also has limitations. Let's think about this for a second. When would you want to administer one versus the other? What are the benefits of broad spectrum medication? Well, if you don't know what you have, right, and the patient needs medication immediately to prevent the spread of that infectious disease. So this is great for immediate treatments. Sometimes you may have had, maybe you have had experience with this in the past, that you are initially administered a broad spectrum antibiotic essentially from the doctor's visit. They give you a piece of paper, or they send it in to your pharmacist. You go pick up a broad spectrum, and then a few days later, you get notification that you need to go and change out your medication, essentially turn in or replace that broad spectrum antibiotic with something else. And what usually happens in those circumstances is that you have your infectious agent has been cultured. That means has been identified, right? And you can, the physician has chosen to narrow down the spectrum of activity to more selectively target that pathogenic agent. So for narrow spectrum is great for, you know, for selectively targeting the pathogen, but you definitely need a diagnosis or determination of etiology. If you recall this from chapter 11, etiology means the causative agent, right? Understanding what that pathogen is that's causing that infectious disease. So because this usually takes a little bit of time, sometimes now we have diagnostic abilities to do very rapid tests for things like strep throat, HIV, obviously that's one. So that allows you to narrow down your therapy, your therapeutic treatment for these different infectious diseases. But things like gastrointestinal infection, those can be caused by a whole host of gram-negative bacteria, sometimes viruses too. But because there's kind of this group of general types of pathogens that cause the same sort of disease, oftentimes culturing results culturing is not even done with those infectious diseases. A broad spectrum antibiotic is administered to try to combat those infectious diseases and kind of left as that. But let's do a very quick reminder of some of the benefits of normal flora, since we're talking about that one here anyway. So let's remind ourselves of some of the benefits of normal flora. And what can happen when you take a broad spectrum antibiotic, right? So essentially some of the things we're listing here, this should just be a refresher for you, but consider that all of these factors here that we talk about are going to be impacted when a broad spectrum medication is chosen. A broad spectrum. So first and foremost, when you take a broad spectrum medication, then you will be impacting gut flora. And remember that gut flora compromises the majority of our normal flora, trillions of bacteria per very short, very small area. In fact, like if you think about it, there's about 10 to the 9th to 10 to the 12th bacteria, like actual colony forming units, so individual bacteria per essentially per gram of your feces. And just think about how much you have in there. Like that's about 1 trillion bacteria for every tiny part of your intestines. So there are a huge amount of bacteria in there. These broad spectrum medications will target those flora as well as others, and so you'll have gastrointestinal disruption almost always. Where you essentially feel the shift in stuff that's happening in your intestines. So these guys are destroyed, they're killed, or they're removed essentially from the protective lining in your gut, and they're flushed out. So because you have, so you'll have, you know, cramping or other gastrointestinal symptoms. Bloating is really common, and oftentimes this leads to downstream problems on the other end of things. So some of the impacts though, remember we talked about colonization resistance.
I also use milk and butter.
Colonization resistance is the benefits that are the benefits that your normal flora provides, right? Loss of, so you'll have a loss of this protective coating. Makes you more susceptible to pathogenic infections. The loss of microbial antagonism. So these are some of the significant factors there. Loss of homeostatic balance.
It's weird. I think our bodies like cycle through.
And that would be pH. That's a huge factor, right? These lactic acid bacteria, for example, fermenting bacteria in your gut producing all that acid that drops that pH, makes it less hospitable to pathogens. So a large factor or a large percentage of those fermenting bacteria will be removed, so you'll have an effect on pH and other factors too. So actually, because of this, there are a couple of really key types of pathogens that are really, really common after the course of a broad spectrum antibiotic. The most notable one is Clostridium difficile. Clostridium difficile, which usually is termed C. diff.
I don't really care.
The C. diff infections are caused by this particular spore forming protection from the colonization resistance of normal flora, but because of the loss of all of these factors here with broad spectrum antimicrobials, and we become super sensitive or susceptible to a C. diff infection, which can be extremely devastating and take many, many years for that to clear out of the body. In fact, we'll hear about this in some of our microbiome presentations coming up here soon. The other pathogen that is really common as an overgrowth of our what's usually kept in check is Candida. Candida albicans, for example, which is a yeast, and this leads to yeast infections in women, which is very common, or thrush, which is a throat or back of your mouth and throat infection in really everyone else, right? So these are two really common types of Candida albicans infections. You can actually get this in your stomach, in your small intestines as well, and it causes some kind of gastrointestinal disruption. C. diff is in your intestines. So these are really, really common infections that can occur after broad spectrum antibiotic medication administration because of the loss of colonization resistance. But remember that there are other factors here too. So regulation of our immune system. Can get out of balance. Metabolism. Remember that our normal flora contributes to maintaining our metabolic activity, producing vitamin K, as well as other basic nutritional components. You guys are doing your microbiome presentations now, so you already understand how significantly the metabolic activity of the microbiota contribute to our own metabolic activity in many different aspects. And of course, cognitive, behavioral impacts, right? So that's the gut, brain, microbiota axis. Right, so each of these things, these are the 4 broad categories that we talked about before. Each of them can get significantly disrupted when you are taking a course of broad spectrum antibiotics. This is not very well studied, but is definitely known that broad spectrum antibiotics can actually have behavioral consequences. So if you're taking broad spectrum medication and you're starting to feel really cranky, it could be a direct result of the altered microflora in your gut and the loss of, for example, serotonin production, so you don't feel very good anymore. All right. Okay. So with these concepts in mind, and I'll have you guys do this during the lecture discussion time, but you'll use these terms to describe essentially the ideal drug. And in some cases, of course, you need to say there are limitations, or under this circumstance, you might want to choose this type of medication or the other. When you're discussing toxicity, you really want to emphasize the therapeutic index here, right? Therapeutic index. Do you want this high or low? And sort of what that means about the patient and the toxicity to the pathogen. Short or long half-life, right? So you're talking about the dose, the concentration, bacteriostatic, bactericidal, targeted to infection, taken orally, right? All of these things are things to keep in mind when choosing the most ideal antimicrobial medication. So the next set of slides are just going to be talking about the basic mechanisms of drug action, so how these drugs work on the bacterial cell and the major classes of these drugs that we have available to us in our therapeutic repertoire. Now, I'm not going to go into this in much detail during the lecture discussion time, and actually not a lot of detail here on this lecture overview, because just like in the last chapter, there is a table that I have for you to fill out in your workbook. So if you go to workbook for chapter 10, you'll find essentially a whole list of different antibacterial drugs and different categories or characteristics that I'm asking for you to complete for these ones. So go through that table on your own time. You can find pretty much all the information in your textbook. It'll talk about applications of the drugs, like when you would use them, limitations of the drugs, if there's toxicity known or contraindications or any other things to consider, and talk about the site of action and how it works. So definitely use that for or use that table to complete, to really get an understanding of each of these different antimicrobial drug categories or classes. And you can use that on your take-home exam. But that'll really just help hone your studies when you're studying for the final, which you will not be able to use that table on. So these are the 5 different sites or mechanisms of drug action. Now note that anytime you see this term synthesis means that we are usually targeting enzymes. This is different from when we were talked about in our last chapter on antimicrobial control methods. Oftentimes we were talking about those antimicrobial control chemicals that are targeting different sites on the bacterial cell and interfering, for example, with cell membrane. Most of these antimicrobial drugs are competitive inhibitors. Which means, if you recall from our metabolism chapter, these guys act as mimic substrates for these enzymes, and they bind covalently, irreversibly to the enzymes, which then inactivates them directly. So cell wall synthesis, the enzymes involved in making the cell wall, protein synthesis, right, the ribosomal complex, the enzymes involved in the ribosomal complex, or sometimes physically binding to the ribosomal complex and preventing the synthesis of proteins. Folic acid synthesis, again, this would be enzymes. Nucleic acid structure and function. There's many different places along this pathway that we have antimicrobial medications to target, sometimes Most of the time they are enzymes, and some of them actually selectively target the nucleic acid themselves. And then cell membrane function. Now in this case, they're going to act a lot like, acts like surfactants from our last chapter. So we'll go through each of these things in just a little bit more detail and talk about some of them and some limitations to them. This is a picture from your chapter textbook. I like it a lot because it does, I think this is from your last edition of your chapter, but it shows you each of these things we just mentioned: cell wall synthesis, protein synthesis, nucleic acid, RNA, folic acid synthesis, and cell membrane, and then lists the broad drug classes, the drug categories in each of these things, like your penicillin, cephalosporins, etc., would be targeting the synthesis of the cell wall, and protein synthesis, either the S subunit or the enzymes involved in these ribosomal complexes are targeted by chloramphenicol, your aminoglycosides here, tetracyclines, etc. Here are some examples. Your quinolones will be specifically targeting your enzymes involved in nucleic acid synthesis. This is Cipro, if you've heard of this before. It's a really common antibacterial medication. Sulfa and trimethoprim. Sulfa drugs and trimethoprim are often given in combination for folic acid synthesis as synergistic drugs, and then your polymyxins fit in here as cell membrane drugs. All right, let's look at some of the drugs that inhibit cell wall synthesis. Apologies for the noises in the background if you hear that. So the concept here, because we're talking about cell wall synthesis, which are enzymes that are responsible for making that peptidoglycan layer, the key concept here, or the big takeaway, is that these drugs inhibit growing and replicating, or I should say they kill growing and replicating bacterial cells, because what you're doing is essentially reducing the growing and developing peptidoglycan. You're not killing it like lysozyme does. So the only impact is with a cell that is undergoing expansion, so internal cell growth, and then cell division to make to make two daughter cells. So those bacteria will then have compromised cell walls and then be susceptible to environmental degradation, specifically through to osmotic pressure. Right. So those are the kind of things to keep in mind. These drugs do not go and kill bacteria directly through just destroying cell wall or cutting them up. They inhibit the synthesis of that peptidoglycan layer, which then affects a growing and dividing bacterial cell and makes them susceptible to osmotic pressure. Drugs that interfere with, these drugs interfere with enzymes required for the synthesis and transport. And again, here's this concept that if you just put these something like penicillin or cell wall targeting drugs on this population, you will not see the lysis of these cells right in that moment. But what it does is, as these cells grow and divide, they're developing more peptidoglycan that now will have weak points because of the inhibition of synthesis. As the cell expands, the cell then becomes susceptible to osmotic pressure, and when water comes into the cell, then the cell will lyse. And so now you can see over time, as this population grows and then divides, then these new cells become super susceptible to lysis for osmotic pressure. Here's a nice picture to show you some of these different sites of action. So bacitracin, for example, interferes with the transport of these sugar molecules across that cytoplasmic membrane. Remember that this is the basic structure of peptidoglycan with these NAG and NAM sugars. Beta-lactam drugs, these ones, and I don't have a picture for you quite yet. Actually, I'll pause this and throw a picture up there for you. But these beta-lactam drugs interfere with the formation of that peptide linkage between those NAM sugars. Here's vancomycin, binds to amino acid side chains of the NAM molecules, interfering with peptidoglycan synthesis, right? So all of these enzymes are great at just interfering the development of this peptidoglycan layer. So here's the three just mentioned: vancomycin, which is great against gram-positive bacteria; bacitracin, which is great, broad spectrum, gram-positive also, but it is highly toxic. This is the main ingredient in things like neosporin that are topical, that also have major warning labels on them. Do not ingest this because it is very, very toxic to the human. And then these are examples of beta-lactam drugs, the penicillins, the cephalosporin, and others. So here's an image of some common beta-lactam drugs. I just wanted to stick this up here so you guys can see this concept of a beta-lactam ring. So there's a whole synthesis pathway to close this ring up, and this is what helps give efficacy for these different cell wall synthesis drugs. So here, for example, are the penicillin. This is actually penicillin, but there are penicillin derivatives that have the same basic beta-lactam ring, this double ring structure. And here is the cephalosporin, which is also a beta-lactam drug with this beta-lactam ring here, and also a double ring structure. So there are variations for each of these beta-lactam drugs, but just keep in mind that they share this common beta-lactam ring. And note here, I put in red, or in red already is highlighted the site of this ring that can be cut by a beta-lactamase, which is an enzyme that some bacteria naturally produce. So let's talk first through penicillin. So penicillin, I've mentioned already, is naturally produced through the fermentation from a penicillium fungal organism. And this is a classic example of microbial antagonism, that this organism secretes out this chemical into its environment to really protect for its own surrounding area. And this was discovered sort of, naturally this culture overgrew, and surrounding this culture, there just became this clearance of bacteria. And the physician or the scientist who was looking at it said, aha, this might be antimicrobial, and began doing experiments and did indeed determine that there is an antimicrobial substance that this guy produces, which now we term penicillin. This was also the first antibiotic that was put into human bodies, into practice, at least through our medical community. Today, we synthetically produce penicillin, although it still can be derived from this natural source. The way that this thing works, it affects those cross linkages by binding to peptidases, which are enzymes involved in creating that peptide bond. There are many uses of this thing, but primarily this is effective against gram-positive cocci, so gram-positive staphylococcus and streptococcus, etc. Some gram-negative bacteria can be susceptible, but remember they have that lipopolysaccharide, the outer membrane, that reduces the impact of these things. This is a picture I just wanted to show you of some of these penicillin derivatives. So here's ampicillin that's commonly used, amoxicillin, methicillin, and other penicillins also. And their variation comes in this R group here. The R group for penicillin looks like this. All of these guys have a circular ring, but have various types of carbon-based moieties and the amine groups on a couple of them. So that's where you would see this variation here. All of these penicillin derivatives share this basic core double ring structure with the beta-lactam ring and the second one here. This is sort of how it looks if you were to evaluate the peptidoglycan structure before and after the application of penicillin for a growing peptidoglycan cell wall, that you still see these NAG and NAM sugars that are interlinked here, but the peptide cross bridges are eliminated because these peptidases that are required to put these peptide linkages are no longer effective. Now there are limitations, there are limitations to penicillin. Specifically, some bacteria have penicillin-specific beta-lactamases, or otherwise just general all-class beta-lactamase activity. Here's a picture of how this works. This is an enzyme produced by some bacteria. In fact, this particular enzyme, the gene that encodes for the beta-lactamase enzyme can be passed from one bacteria to another through conjugation, potentially through other means like transformation. So this enzyme here, beta-lactamase, will bind to this beta-lactam ring and cleave the beta-lactam ring right here. But there are ways to inhibit this beta-lactamase enzyme, right? This is what provides resistance or drug resistance to penicillin. It's a very widespread enzyme now, or gene, that's passed throughout many organisms in the population that now have resistance to penicillin. And because of that, oftentimes what you'll see if a physician administers penicillin, they'll naturally do penicillin plus clavulanic acid. Clavulanic acid, or clavulanate, looks a lot like penicillin at its core structure, So this essentially binds to the beta-lactamase, but it inactivates or reduces the ability for beta-lactamase to do anything else. So it essentially like sops up the beta-lactamase. It's not a good way to put it, but it kind of like that, which reduces the beta-lactamase activity against penicillin. Therefore, penicillin can then go and do its action against bacteria and still be effective, right? So clavulanic acid is what you might see as a co-administered molecule in addition to penicillin, or even in addition to the derivative drugs like methicillin or any of the psyllin families, because as you saw, they have the same core structure, so the beta-lactamase enzyme can deactivate those ones as well. The other category here, protein synthesis drugs. These ones, there are many of them. They all interact somehow with that ribosomal complex. And a side note here is that there is potential impact on eukaryotic mitochondria. If you recall, the eukaryotic mitochondria is very similar in function, but it's different in size. No, I'm sorry, the eukaryotic ribosomes are similar in function, but different in size. Our ribosomes are 80S versus eukaryotic or prokaryotic ribosomes are 70S. But again, if you recall, our mitochondria has essentially the same thing as prokaryotic cells, so they have 70S 70S ribosomes. Here's a picture from your textbook, or at least the previous version of your textbook, show you lots of different sites on that ribosomal complex for these five major protein synthesis classes of antimicrobials. So here's your aminoglycosides. These will bind essentially to the mRNA component of this ribosomal complex, which causes misreading and incorrect proteins. Here's chloramphenicol. This one prevents the formation of that peptide bond from P to A, so it eliminates the development of that protein. This class actually inhibits the formation, the formation of that ribosomal complex altogether at that ribosomal initiation, so preventing protein synthesis. Tetracyclines up here in this corner inhibit the binding of tRNAs at the aminoacyl or acceptor site here, and erythromycin will physically bind and covalently prevent that ribosomal translocation to prevent the development of that polypeptide strain. So some of these specifics here, we have tetracycline here. It's a broad spectrum. You probably have taken it at some point in your life. We just saw that this guy binds to the ribosomes, ribosomal complex, and prevents that protein synthesis. It's used commonly as a broad spectrum, so it really has a great impact against a very wide range of organisms, gram-positive, gram-negative, different shapes, aerobic and anaerobic organisms, commonly used in sexually transmitted infectious agents, Lyme disease, even acne as a topical. This is low cost, easy to administer, but of course it's broad spectrum, so it can cause some significant GI disruption. So these are good questions to just sort of think about this. Why might a drug that targets the cell wall like penicillin be a poor drug choice for a gram-negative infection? And why might a drug that targets the bacterial ribosome be a poor drug choice for a pregnant woman or a young child? We'll review these questions in our lecture discussion time. The third category of drugs, folic acid synthesis drugs, right? So these are enzymes, or these are molecules that act as competitive inhibitors or metabolic analog. So sulfonamides and trimethoprim are two classic examples that are usually given as a synergistic application. Both of them are given at the same time because they impact this folic acid synthesis pathway at multiple spots. So as mentioned, this is a pathway that's important for the development of amino acids and nucleic acids. So without this, then bacteria cannot produce these things and therefore cannot grow. The great thing about these classes of drugs is that they're selectively toxic. So we humans who don't have a folic acid synthesis pathway, we have to get our folic acid through our diet because we don't make it ourselves. So by providing this to a human who is infected, ideally this will just target the pathway in bacteria and not the pathway in humans. As mentioned, these sulfa and trimethoprim, these are usually given synergistically, and they have a wide range of applications. And their mode of action obviously is, as mentioned, they inhibit those enzymes. Nucleic acid drugs, again, this is a very long pathway for the synthesis of nucleic acids. So there's lots of different ways that this pathway can be inhibited, blocking nucleotide synthesis, inhibiting replication of DNA, stopping the process of transcription through the elimination of RNA polymerase binding or RNA polymerase activity, inhibit DNA synthesis, right? So all of these different things are target sites for some of these nucleic acid drugs. A common category here or class of drugs is rifamycin. This is naturally derived from a Streptomyces bacteria, but also can be synthetically made. These guys, rifamycin will block the RNA polymerase from initiation and is a bactericidal, so it actually kills the organisms, obviously because they cannot make proteins and therefore all metabolic activity is halted. This has limitations. It cannot pass through that gram-negative cell envelope. That lipopolysaccharide is inhibitory. So it's very effective against gram-positives and really great at use against mycobacterium. Fluoroquinolones are in this category as well, which are inhibitory towards its broad spectrum, so towards a large class of organisms. This is your Cipro or moxifloxacin are in this category as well, These are RNA replication inhibitors, so blocking that topoisomerase so that replication cannot ensue. This last category are the cell membrane drugs. As mentioned, they kind of act as surfactants, right? So they disrupt that cell membrane, lead to the permeability of that cell membrane, which leads to inhibition of metabolic activity. If you recall, all respiration pathways are based on that cell membrane, the integrity of the cell membrane. So inhibiting or disrupting the cell membrane can lead to halting of metabolic activity through that way, and inevitably lead to cell death. This can be species specific because it inhibits or it interrupts or interferes with the lipids. But the unfortunate part is that although you can get high selectivity here, the lipids do have some similarity to human lipids, and so it can be toxic to humans. Polymyxins are in this category, derived from a bacillus. It is narrow spectrum because of its selectivity of lipids. Again, it kind of acts as a detergent, so a surfactant, and it does have significant limitations. This is usually used as a last choice drug at compassionate care. If you have a drug-resistant organism like the super common Pseudomonas aeruginosa, which is a nosocomial agent, or some gram-negative rods for urinary tract infection. This is another category, a very similar concept here of this drug inserting itself in the cell membrane, used really only against gram-positive bacteria. So here's some good, just broad discussion questions. Actually, one we've already talked about, so you don't have to do this one again, but you've been given the job to create the ideal antimicrobial. What characteristics should this drug have? Really, that's the same kind of answer as you gave in the very beginning of our lecture slides. This one here, again, just a broad abstract idea. A new chemotherapeutic drug kills bacteria but not humans. Discuss one possible way the drug may work to target only bacterial cells and not human cells. In fact, this is a great opportunity for you to go back to chapters 3 and 4. That chart that we were creating that hopefully you guys finished, where we were comparing and contrasting prokaryotic and eukaryotic cells and really identifying the different composition to different components of these organisms. Just go back to that chart and do a nice review to really select out the components of prokaryotic cell that would be a really good target, for chemotherapeutic drugs. Here's a third question. Your pregnant neighbor has been prescribed a daily dose of oral tetracycline for acne. Why do you think this therapy, or do you think this therapy is advisable? Why or why not? Another good discussion question for you. A woman has been prescribed a broad spectrum oral cephalosporin for strep throat infection. This is really common. What are some possible consequences in addition to curing the infected throats? All right, and that's it for this first set of slides. We'll do part 2 in the next set.